Nezmi Alem Vakıf University, Istanbul, Turkey. Thank you for joining my session. My talk is on a study that we did during the early days of COVID-19 pandemic. And we wanted to identify and characterize neutralizing B cell epitopes for SARS-CoV-2 when we did not have any such data available yet. The spike protein of SARS-CoV-2, which mediates fusion and entry of the virus into host cells, is a primary target of the host immune response and could provide insight into the selection of vaccine targets and immunotherapy. In this study, to reiterate, we aim to identify and characterize a neutralizing B cell epitope on SARS-CoV-2 spike protein. Specifically, this study was done in April 2020, and when there was a lack of reported information on experimentally verified neutralizing antibody epitopes against the S1RBD of human SARS-CoV-2 spike protein. Those known neutralized strains of the earlier close homologs, SARS-CoV-1, responsible for the 2003 outbreak, was used as a proxy to identify the neutralizing epitope for SARS-CoV-2. We also wanted to study the diversity of the epitope sites that we would identify. The methodology workflow is shown in this figure in here. We first retrieved relevant 3D structures of SARS-CoV-1 complexed with a neutralizing antibody. We also downloaded spike protein sequences available in public databases. The retrieved 3D structure was processed to analyze missing residues and the assessment of the resolution quality of the structure. Whereas for the primary sequences, we removed the duplicates, aligned the resulting non-redundant sequences, and corrected for any misalignments. We then analyzed the selected 3D structure to map the neutralizing antibody epitope on the SARS-CoV-1 and the corresponding prototype on the antibody. The identified epitope sites were then mapped to SARS-CoV-2 through a alignment correspondence. The map sites were then analyzed for their diversity by the use of this alignment. So this was the workflow we used. The PDB record 2GHW the X-ray resolution with a 2.3 angstrom was used as a SARS-CoV-1 structure for our study, and the record provided a crystal structure of SARS spike RBD in complex with a neutralizing antibody ATR, and ATR is a neutralizing human monoclonal antibody blocking the binding of S1 to ACE2 with nanomolar affinity and preventing the formation of the syncytia and inhibiting the viral replication. As shown in figure 1, in figure 1, the molecular visualization systems, PIMOL and VMD, visual molecular dynamics, were used to identify the neutralizing antibody epitope residues on the SARS-CoV-1 subunit S1RBD, which were recognized by the residues of ATR within proximity of 5 angstrom distance. As for the spike protein primary sequences, they were collected from the virus the VIPR, NCBI virus database, and GIS8 databases as of June 2020, and duplicate sequences were removed by the use of the tool CDHead. And let's dive into the results. So this table 1 shows the epitope and paratope residues determined by the different visualization systems. In red are the overlaps between the two systems. In here, VMD provided several extra residues, and upon further selection, we ended up with a total 99 residues of the S1RBD that were observed to be interacting with 34 residues of the neutralizing antibody in the PDB structure in 2GHW. And these 29 epitope residues and the 34 paratope residues are shown in this table 2. And in here, so the distance in angstrom is shown in the brackets in here. And the 29 neutralizing antibody epitope residues of S1RBD for SARS-CoV-1 were correspondingly aligned mapped to the SARS-CoV-2 spike as shown in table 3 in here. And these mapped neutralizing antibody epitope positions were visualized on the 3D structure of human SARS-CoV-2 S1RBD spike as shown in figure 2. So in here, and this one to assess their structure, localization, and exposure to binding. The putative neutralizing antibody epitope sites on SARS-CoV-2 spike were then analyzed for their diversity in the reported population of the virus. And removal of duplicate sequences from the initial data set that was retrieved from the various databases, and 2,240 non-redundant SARS-CoV-2 spike protein sequences remained. And these sequences were used to analyze the amino acid variability among the strains of the virus for the epitope positions corresponding to the paratope of the ATR neutralizing antibody. And the results are shown in this table in here, table 4. Only 7 of the epitope positions showed variability. However, these sites were restricted to a negligible fraction of the viral variant sequences, and the minimal variability of the neutralizing antibody interacting residues observed in agreement with the reports of high efficacy of vaccine that target the spike protein. In conclusion, the approach herein demonstrates the value of existing 3D structures of neutralizing antibodies in complex with a virus protein for identification of putative neutralizing epitope residues for a novel homologous virus protein. Thank you, everyone.